I'm Jennifer, and my slides are there. And this is Brett, and we're both from Solventa. So just to give you a background of Solventa, we're a diagnostics and consulting company. So we work with pharmaceutical companies as well as consumer products companies to help analyze their big data. And we use our biological expression language to compute on the biology in their data and to predict upstream molecular regulators of mainly transcriptomic data that we work with. So BEL is a great way to do this. It's a computable language, it's human readable. Brett's going to give a background about BEL, and then I'm going to talk about using BEL for crowd verification of biological networks. So this SBV Improver is a group that actually helped sponsor this event. And we were hired by this organization, which is made up of, sponsored by Philip Morris, Orange Bus, and ADS. So it's a collaboration of many different companies. And what it is a organization that tries to use crowd verification for verifying different systems biology techniques. And it's in its third year, and this is the Network Verification Challenge. But there were a couple other challenges before, a Diagnostic Signature Challenge and a Translatability Challenge. And I'll give a brief background about those challenges. Those were all contests that were put out by the crowd, similar to the DREAM Challenge, where people were working on a specific set of data and trying to come up with an algorithm. This is a little bit different, where the crowd can go and look at these networks that are composed of these BEL statements and contribute their biological knowledge. So we're using different expertise gathered from the crowd to try to make the best biological networks that can then be used in biological research, computational research, and we're actually going to give a webinar more on the applications of these networks in a few weeks. We'll put the date up. Brett will be giving that webinar. So just to give an overview of what we'll be talking about, the BEL language, then the Network Verification Challenge, and then the BioNet website where you can see these networks and you can also download the networks, use them for your research, and then we'll have questions. So this is just a graphic to show you how this all connects. So these networks are composed of nodes and edges, and they're scripted in BEL, the biological expression language. And Brett will go through the value of BEL and the type of metadata that you can attach to each statement. And then I'll go through the networks that are on this website that you can help verify by contributing your biological expertise, or if you know other people who are in biology, then please let them know about this challenge. One of the incentives for participating is that next year in June 2015, we're going to have a meeting in Barcelona, Spain, and the most active contributors to the website get invited. And here we talk more about the biology of the networks and make decisions on how to restructure these networks and how to, what things are missing, what things are controversial. So it's a chance to meet in person where things can't always be figured out online. So it's a good chance to network with other experts in the field. We invite subject matter experts as well, and the people who created the networks. So that's coming up next year. Okay, so I'll hand it over to Brett to talk about the BEL language.
Okay, so let's start at the ground level and just basically give an overview of what the BEL language represents and how you can use it in your own scientific research. So essentially the idea behind BEL is to capture scientific findings and essentially publications in the public domain to represent these findings in a human readable but yet computable format. So as a lot of you are probably aware, so you start with the scientific literature, so you can do a PubMed search and find any number of scientific publications on any number of topics. And then what you find from that is essentially causal relationships in that literature. So one example here would just be the RNA expression of RBL2 is directly mediated by the activation of the FOXO3 transcription factor. And this has an associated citation information and PubMed ID associated with that. And what you can do is essentially represent this plain text statement or causal statement as a BEL statement. And so when you convert this into BEL, you essentially have this statement where the transcriptional activity of FOXO3 directly increases RBL2. And so I'll go into the details of exactly how you essentially parse out each of these segments of a plain text evidence quotation from a literature, just from a piece of literature, and then construct a BEL statement from that. Yeah, so basically what differentiates BEL from some of the other languages in the scientific community, especially in the biological sciences. And so BEL represents discrete scientific findings in a computable format by capturing qualitative causal relationships and irrelevant contextual information. And so what I mean by contextual information here is essentially when you read a scientific paper, it has any number of metadata qualities attached to that. So when a lab does a given experiment, that can be in any number of cell types or tissue subtypes. And so what you would like to do ideally is capture the causal relationship that they actually found in that scientific study, as well as any of the associated metadata. So did that occur in the context of lung cancer? Did it occur in a specific cell type? And so all of this with BEL you can capture within that statement.
But actually, biopass captures also tissue information.
Yeah, I'll go into that.
Experimental evidence.
Yeah, I'll go into that on the next slide. And I'll also go into the fact that BEL can drive knowledge-based analytics. And as you just mentioned, there are other languages in the field, certainly BioPact and SPML are the more popular languages. And basically these other languages are designed primarily with pathway construction and visualization in mind. So they do capture causal biology to an extent, but at the same time, it's really difficult to annotate that with specific literature metadata that are captured with that. And so with BEL, you can really focus on, just focus on the detailed molecular level as opposed to all levels, including general process level. So with the more specific languages like BioPact and SPML, you can really represent different forms of a protein in a causal pathway, and essentially, just focus more on like very, very fine detailed processes, whereas BEL is really meant to represent more the process level causal biology in a given relationship. And another fact is these other languages don't necessarily capture this contextual information that I just mentioned. So what I mean when I say that BEL is a computable language is basically just the fact that you can construct biological networks using BEL that can be scored with any number of different omics data and facilitate more of a systems level understanding of what's going on in that data set. And so just to give a brief example here of some type of experimental perturbation that someone may do in a lab, so you can have any number of like knockout animals, you can have chemical perturbation or any kind of pharmaceutical treatment here. And then what you get from that data is when you can do transcriptomic profiling or any kind of protein ELISA or any kind of any endpoints in a scientific study to get this systems biology level data. And then from here, this is where the networks can come in that you build using BEL. So at this level, we have different networks that can represent any number of processes from cell proliferation or cell cycle or climate inflammation, for example. And what you can do, and a lot of these techniques are freely available as well, is essentially use these networks to score exactly what's going on in those data sets at a more high-level systems biology level and get a number of different quantitative outputs to give you a better understanding of like condensing that gene expression data down and get a more high-level understanding of what's going on in that data set. And so here's to your point more about comparing directly between BEL and BioPact, for example. So on the left, we have a representation of a BEL pathway in a network. And on the right side, it's more of a prototypical network that we pulled out from the BioPact language. And so when we have BEL, essentially we have this causal relationships backed by discrete scientific findings with very specific scientific context. And so if we focus on this example here in the BEL pathway, so we have the kinase activity of IRAK4, increasing the kinase activity of AKT1, for example. And so just to go back to this idea that in BEL, each causal edge is supported by a specific publication and annotated with contextual information. And so in this specific example again, so here we have the statement again, the kinase activity of IRAK4 increases kinase activity of AKT1. And so from this associated PubMed ID that we pulled out of the literature, we have the species context, the cell types, so in this case neutrophils, and a specific just a reference literature in the quotation that supports that underlying causal connection in a network. But if you look at more of a BioPAX network here, so this details more the activity of the AKT1 protein. And so what the BioPAX language does is essentially allows you to very, very precisely annotate different states of the AKT1 protein. And so if you start with the base case, I think it's here at the top. So AKT has 2 possible phosphorylation sites, for example. And so you can capture really how these different proteins interconnect, and you have multiple phosphorylation states derived from this like sort of basal state of the protein. So here it just represents that one site is being phosphorylated at this location. And then it goes on to show like just more precise information about the AKT1 can also be phosphorylated at both sites to mediate a specific biological function. And then you can also capture essentially complex formations when you have certain states of the protein. So here in the AKT1 example, if it's phosphorylated at both of these sites, we have a complex formation with HSP90. And so this really is useful for giving an overview of exactly precise processes surrounding a single protein, but at the same time doesn't really give you causality and form very, very full functional pathways in a network, for example. And so going back to BEL, we can very clearly illustrate in a network type architecture that this causality between IRAK4, AKT1, and increasing this other protein in a very systematic way. And each of these edges in this pathway are annotated in the same way. So we have an associated species, cell type, element ID associated with each of these.
I'm sorry, but first of all, the last sentence where you say all pathways may be annotated among specific statements in the pathway. Like in BioPath, you can annotate every single process. So I don't know what it is that you want to annotate, but can.
I mean, what do you mean, like the specific connectivity between these components? That you could...
Well, it's connected by processes. Everything, like every interaction in BioPaths can be annotated.
Yeah, but it's not annotated with specific literature backing that edge. So the edges in this network don't mean anything. It's just to say that this interconversion between different states of the protein. I mean, each of these edges is not supported by the literature. Like
it's the process, the whole process that's supported by literature. Like
right.
Every chemical reaction you have in that pathway, you can have annotation in literature and cell types and so on.
For each of these edges, you mean?
No, but the processes that connect the entities. Like if you have one protein interacting with another protein, there's an interaction node that describes interaction. That node can be annotated.
Okay. I see. I mean, we do have a slide later to give a more systematic comparison of like how the languages compare. So we can go through that at the end if you want to, if you have any other questions. Okay, so essentially just how to decode a BEL statement. So a BEL term, or a node as it's represented in a network architecture, always appears in the following format. And so you have a given node in a network, and each of these functions within the statement are represented here. So first we have an associated function with that term, and we also have a namespace, which is a public namespace that contains a gene symbol, for example, in this case HGNC. And we also have the entity term, so AKT1 is the specific protein we're referring to in this case. And so if you break this down a little bit further, what we have here, if you start from this end, for the entity term, this is a biological entity of interest. So, of course, this could be any gene or protein that you're interested in, as well as a biological process. And then in terms of the namespace term, this represents a public database containing this entity. So what the language is able to do is essentially dynamically go back and associate it with these public databases to always have the most updated terms available if those ever change. And then here at the function level, this essentially just describes an abundance or a process related to this entity. So it could just represent the protein abundance, for example, but it could be some kind of catalytic activity or kinase activity associated with that protein. And I'll go through a lot of examples for each of these in the next few slides. And so here's just a slide that gives an overview of some of the possible functions that are associated with the BEL term. And so we have any number of functions here, so abundances, modifications of proteins, processes, activities, transformations. And this just gives sort of a prototypical overview of an example, again, related to the AKT1 protein. So if we start here in the middle with just the base protein abundance function, so this is represented by P, and again here we have the namespace HGNC and of course AKT1. And the way the other functions have been represented here is essentially you can do protein modifications, and so the protein abundance function here has this additional segment here to indicate a protein modification. So phosphorylation at 3NA308, for example, and then phosphorylation at serine 473. So these are just ways you can represent the base modifications of existing proteins. And then further from that, you can use these terms, group these terms, these function terms together to represent sort of more advanced processes related to this protein. And so here's the case of activity, so the kinase activity at AKT1. So again, you would start with the protein abundance sort of base function here, so P, HGNC, AKT1, and then you could wrap this in using the kin term. So this represents the kinase activity of this protein. And then here's just a causal connection demonstrating that the kinase activity of AKT1 increases the process of T cell activation. So here, it's a process is represented slightly differently, so we have a different namespace here. So the gene ontology namespace, as well as the BEL function, which indicates a biological process. And then here's just to give a little bit more of an overview of the possible abundance function types that are possible within the language. So basically, this can just specify the presence of an individual RNA or protein entity, the symbol of which is derived from the associated namespace, which I mentioned. And the more complex term is the complex function, which uses to combine multiple abundance values into a singular molecular complex term. And so a lot of these possible functions are abundance, protein abundance, which are pretty obvious. And you can also combine multiple terms into this complex. And we also have functions to represent gene abundances and microRNA specifically, as well as just RNA abundance of a given species. And then in terms of activity functions, some that are possible within the language. So these are just applied to the protein and complex abundances to specify specific molecular activity. So these can be any number of possible biological activities, ranging from catalytic activity and GTP activity. So these are represented by pretty obvious symbols here that precede the namespace in the protein. So again, we have kinase activity examples and peptidase activities, phosphatase activities. So any of these common biological processes can easily be represented as discrete functions within the language. This really gives a lot more granularity to how you can represent different protein states within a given network model, for example. And here, just to give one more slide on possible functions within the language. So these are related to modifications and some sequence variances for proteins. So this can include anything from phosphorylations, for example, ubiquitinations, as well as specific mutations and polymorphisms encountered within a protein. And so for protein modifications, these get a little more complicated, but you can very easily represent phosphorylation states of a protein, acetylation states, and substitutions that are common in the literature. So substitution for these amino acids, and you can also represent fusion proteins here as being specific chromosomal sites within those fusion entities. And when it's related to process level functions, this really just includes more higher level biological processes that you can represent within the language. And so biological process is by far the most common term we have in our networks. And this is just represented by the BP function. And this can represent anything from cellular senescence to apoptosis to T cell activation, any number of things. And so all of these are captured within the gene ontology public database, and I'll go into a few more details related to that in a few slides. And we can also represent sort of pathological processes using the language as well. So this very often derived from the MESH database, and so you can represent pulmonary disease, chronic obstructive, so COPD can be represented as a discrete term within a biological network architecture. And then moving on to the next part of the structure of a BEL term is essentially just the namespace, which we've gone through a few examples, but just to give one very common example that appears in our networks is the HGNC namespace. And this is essentially just the Hugo Gene Nomenclature Committee database. It's a collection of essentially all known human loci, so this includes over 37,000 loci, of which 19,000 are protein coding. So this really is a very, very comprehensive and dynamically updated database that always has the most current gene symbol, and the BEL language is capable of going to that database and pulling those symbols out. And so as I mentioned, most nodes in the networks will be derived from this namespace just because for the most part, at least in our research, we deal with human proteins, and so this is sort of the database of choice to go and find those specific gene symbols that you would represent as a BEL term. And then another namespace here would just be the gene ontology database. I'm sure a lot of you are probably familiar with that one. And this just represents more common biological processes like neutrophil chemotaxis, for example. And so again, this namespace is just derived from the public database for gene ontology that's updated continuously. And so, this could just represent any number of processes. So you could search for T cell activation or apoptosis, for example, and just get a specific gene ontology term that you could use and map as a BEL term. And then just related to how to decode an edge within a network architecture. So the way these BEL terms are structured is, again, you just have a function, a namespace, and an entity. And this is represented as a discrete node in a network. And so based on some published experimental finding, BEL terms are connected together using a relationship to specify a functional BEL statement. So basically the process of connecting one BEL term with another BEL term forms a complete BEL statement here with this relationship. And so to go into a specific example for this, if we look at the catalytic activity of caspase 1, for example, increasing the protein abundance of IL-1 beta. So what we have here is essentially multiple evidences that support this edge that have been derived from the public literature. And if you look into what you can do within a network structure is basically just look into exactly the literature reference that supports this edge, as well as an associated species and cell type associated with this. So all of this metadata is captured within a specific BEL statement. And Jennifer's segment, she'll go into a lot more detail about how this is organized within the network verification challenge and sort of the networks on the Bionet website. And so here is just how you can represent different relationships within a BEL statement. So one of the more obvious ones, of course, is an increases type relationship, which is, I would say, forms about 80% of the relationships that we've modeled in our network so far. And so this is just represented by this pointed arrow here connecting these terms. And then a decreases related type relationship is represented by this ball and stick sort of component. And directly increases, you can visualize as more of a dark black arrow here. And then directly decreases, we send this dark black ball and stick connecting these two BEL terms. And then I would say between those four, that represents like 90-95% of the relationships in our what we call our knowledge base, which is a collection of all the BEL statements that we've curated over the years. But one of the more common edge types is this non-causal type relationship, which is represented by this dotted line. And so what this means is essentially there is no literature that supports this edge, and yeah, there's just no underlying literature reference. And so this really just relates to specific protein to activity type connections. So going back to the AKT1, for example, that I showed, if you have the central feature of a network being, or one of the nodes in the network being the protein abundance of AKT1, and you have multiple phosphorylation states of that protein, you would represent those connections as non-causal edges. And so there wouldn't be a specific reference supporting that edge, but you can still represent it in the network. And so basically moving on from just the idea of BEL terms and BEL statements in general. So collections of BEL statements can then be used to assemble biological network models. And so we have this just generic network architecture visualized here that we use, well, in our research we use Cytoscape quite often to do the visualization. And the way these edges are represented here is, just one term connected to another using any number of these causal type edges. And then for each of these edges in the network, we have a specific literature reference and quotation, and the specific content and the context annotated as part of the edge as well. And so in most cases, the networks are supported by literally thousands of peer-reviewed scientific findings, and each edge in the network can be supported by multiple findings. So this could, for this edge, for example, in the context of human Th2 cells, this relationship could also be found in cancer cells, for example, and that would also be included as part of our knowledge base that underlies this network. And then moving on to the idea of just more of a network-based architecture, and then network models that are constructed using the language can really be used for any number of applications in pharma research, so drug discovery, biomarker discovery, and more toxicology type applications. And I think Jennifer will go into a few more examples of this in her talk as well. And then so with that, I would just wanted to list up here a few more public resources that have been published that can familiarize you with a lot more of the intricacies of the BEL language if you're interested. And so one of the primary resources would be the Open BEL Consortium website. And so this is updated by Selmensa and also a number of our collaborators. It gives a really great overview and has a lot of good presentations to give more of an overview of the language and how you can apply it to specific applications in your own research. And the Open BEL Wiki, of course, is updated all the time with new information, and a lot of users have gone there to post specific examples of how they've applied this in their own research. So that's a really good resource that people can check out. And also another interesting resource that's been deployed as part of the network verification challenge that Jennifer will also talk about is this plain text to Open BEL text converter tool. And so what you can do with this is essentially take an abstract from a scientific finding in PubMed, for example, and just input it within the text field, and you can submit this and it'll send you back BEL terms and BEL statements that can be derived from that abstract. So it's a really useful tool to just get up to speed quickly about how terms are converted into Open BEL language. And then of course the GitHub repository, we have a ton of documentation and downloads for BEL framework and other tools that you can use. And also we have a sample of pre-compiled CAMs, which are knowledge assembly modules. And these are basically huge collections of BEL statements that we've curated over the years. And these are all freely available for you to take a look at and potentially apply in your own research. And then of course, if you have any specific questions, you can ask now, or of course feel free to email me at any point. So I guess with that, do you guys have any specific questions about BEL or? Yes?
Yeah, so for example, if I have in my knowledge base a statement that says A increases B, and the second statement that says B increases C, does it give me license to conclude that A increases C?
I would say it depends on the context, because more often than not, I mean, the underlying relationship between A and B may not be from the exact context or cell type, but the relationship between B and C.
Let's say it's the same cell type.
So yeah, I mean, at least as far as we've built our network models in the past, I mean, yeah, that's what you would conclude.
It's a model, so you go to the literature data, you would say that's the model.
Well, it's the same literature.
Yeah.
I mean, everything has to
This
be
is only
experimentally
true of
validated. These are all theoretical models, so it's a possibility that A could increase C, but it's also possible that it doesn't happen. So you of course have to validate that experimentally. But these models are built to give possibilities for researchers to, to open their eyes to maybe areas of interest that they would want to investigate further.
So the purpose of BEL is like to help you make sophisticated guesses?
Yeah, it's a way, for in our company we make these hypotheses based on the data, and it's a way to describe biology in one type of network. It's definitely not something that is set in stone that this has actually happened. So it's a theoretical framework that scientists work with, and it's also a way to share data, because a lot of this is built on data. For our customers, they have data that we then use to build these networks. And so the great thing about BEL is then you can share the different biological theoretical frameworks between different groups of people or different labs. So trying to make it a standardized language for people to share information. Because in biology, there's all of these literature references, and it's really difficult to read hundreds of references. So we're trying to compile it all in one type of language.
I think to push the example of the challenge, you can also often find a reference that says A equals A increases B, and another reference that says A decreases D.
Exactly.
And so it is messy.
But there is no inferencing, which is what I thought Alvaro was asking. There is nothing built in here that assumes certain inferencing rules. It's strictly you model the data as you know it. There is no rules about if A increases B and B increases C, therefore A increases C.
Yeah.
There's nothing like that built into the language.
No, it's not built to that. That's something that you can create as a researcher. You can overlay your own tools and
With
algorithms.
With what?
With your own tool. A lot of people build their own kind of algorithms to use these networks. But like you said, there's nothing built into.
But is it easier than just to extend the RDF model, because BEL is ported into RDF, right? We're able to proper RDF triples, and then you could potentially add actual semantic-based reasoning to that.
Yeah.
So I think that's really interesting presentation in framework. Just a couple of questions. One of the things that drives us all crazy is naming and naming convention. And so you spoke about Hugo names. Is there a sort of lookup table and a translation table that you guys have built, such that if someone calls it Mick 1, someone else calls it build that into here? Or
Yes.
is it all user input?
No, so as part of they basically list the official meaning, but they have all associated synonyms with that as well. So if the user types in a synonym, it will give you the official term to use as a BEL term.
It will translate.
Yeah.
And it's so important.
Yeah,
That's the hardest in literature is everyone, even today, people ignore the Hugo name and use the name that they published on last.
Yeah.
Yeah. So that's nice. And, another thing is there's probably in here, but there's sort of emerging, powerful, massive data set of SNPs and EQTLs is coming online, and it's often not so much in the papers, but you can look it up. And have you guys built SNPs, which of course are dispositions and often not exactly gene associated? Have you built any of that framework, which is going to be so important for understanding human validation, human risk, human variation?
Yeah, that actually came up recently. We just submitted a paper and someone asked about that, and that's something that we're developing. So the next version of BEL, right now we're on BEL 1.0 or BEL 2.0. So the next version of BEL we're trying to incorporate the SNPs, but currently we don't.
Based on looking at sort of new targets and validation, getting out some cell line system going into human populations, so much more powerful than the context of some cell running culture. And so it really promises to be useful, but it's really a mess right now and hard to interpret. So building that into a language could be really hard. Another question. So you mentioned AKT as an example. I mean, AKT is going to have 10,000 papers on it, AKT1.
Yeah.
So what does that look like? Are you free to ignore? I mean, do you then build a database and you have 100 or 200 things that AKT1 does?
Oh yeah. That's part of our, what we call it the knowledge base.
So, I mean, that must be a mess, right? What does that look like as a network when you've got 100 different
I mean, so usually when we build
things
network,
that AKT does?
yeah, so usually when we build a network, it's essentially like a discrete cell that we're trying to model the pathways in. So
Or
if we-
disease, or tissue, yeah.
Yeah, or tissues. So if we want to model the specific processes related around AKT1 and neutrophils, for example, I mean, it's super easy to go in our knowledge
Yeah.
base and pull out those statements.
So you ignore everything else that does, which for your context is.
Right.
Yeah, that's a great thing of having everything annotated that, if we're looking in a specific disease or a specific tissue, then we can just filter out what we want and put that in the network. We don't put everything associated with AKT1. We only put what's relevant to the disease we're modeling.
Yeah, because AKT1 and T cell activation is probably like the 100th most important thing that AKT1 does.
Yeah, exactly. Okay, so I'll hand it over to Jennifer to go through the network verification.
Okay, so now that Brett introduced BEL, I'll talk a little bit more about the SBB improver and one of the applications where we can use these networks. As I said before, we have pharmaceutical clients where we analyze their data and we build different networks for them. In this case, we built 50 networks for one of our clients who then wants to verify these networks through this SBB Improver challenge. And so why do we need this SBB Improver organization? Well, there's a ton of data out there, and there's a ton of ways to analyze the data, but how can we validate that? There is the peer-reviewed literature process we go through, but a new emerging way of sharing and collaborating is online and through crowdsourcing. And so this is one approach that SBB Improver is using to try to validate robust methodology for system biology-based approaches. So SBB Improver stands for Industrial Methodology for Process Verification and Research. And the main concepts are to formalize rigorous tests that determine a decision for a systems biology research pipeline in an industrial context, to inspire the development of enhanced methodologies by community participation, and to endow the community with data sets and benchmark to provide a means for continuous improvement. So for the first two challenges, people were working on the same data and coming up with different methodologies. So you could always compare side by side these methodologies if you're working on the same kind of data. So to go through some of those challenges, the first one was the diagnostic signature challenge. This took place in 2012. And this was a challenge to be able to come up with a signature, a gene signature of disease and normal. So there were many different types of data sets. There was a psoriasis data set, there was a lung cancer data set, and different teams around the world competed to try to come up with the best signature that divided disease versus normal or divided different subtypes of lung cancer. And there was a winning team, and they published that algorithm in a publication. Similar to that, the species translation challenge, where it was another challenge looking at the same data, so rat data and human data. How well does these two species translate? What kinds of things in human can be translated to rat? And this was another kind of algorithmic approach that people took. And there was a winner, and people published their algorithm. And you can find this on the sbbimprover.com website. And then this third challenge that's ongoing is the network verification challenge. So we had NVC1, which was the first challenge last year. And that ended, the open phase ended in February. And then the NVC2 is ongoing now, so you can contribute currently, and that will end in April. And then we'll have the in-person meeting. So I'll go through kind of the steps of NVC2 and how we went about the different phases. So just to give an overview objective, the objective of the MVC aims to verify and enhance these biological network models. And then these models can then be used for drug discovery, personalized medicine, and toxicological risk assessment. The first NVC took place last year, closed February 2014, and participants reviewed the biology, they added new biology, and it was all based on their expertise. So it was a great way to just pool everyone's expertise together on one website, looking at common networks. And as I said, this NVC2 is ongoing, ending next April. So the different phases of this challenge. First, we constructed the models. So Solventa created these models over the past couple years. We published these 5 publications in different areas: proliferation, cell stress, inflammation, DNA damage and cell death, and tissue repair. So it covers a wide variety of processes. There's about 10 networks in each of those subject areas. And we built that both on literature and on data. The second phase is this online open phase, where you go to this website, you review all of the different edges that are in the BEL format, and you can comment, you can vote, you can add your own biology, and interact with people that are also participating. But there's a community page where you can see the most recent activity. So this is really the bulk of the challenge, where people contribute online. People who wouldn't normally collaborate together are brought together by similar interests in a similar subject area. So it's a great way to meet other people in your particular area of research. And then the next part of that is this jamboree, what we call a jamboree, which is an in-person meeting, which we had in Montreux, Switzerland last year. And we brought together the best performers, so the top 20 participants who were most active and contributed a lot. They were invited to this meeting with subject matter experts and with the scientists who built these networks. And we all sat in different rooms according to our subject area expertise. So we had a DNA damage network session and a neutrophil signaling network session. And we really worked in these small groups and discussed, how can we represent the biology of neutrophil signaling in the best way? What were some of the contributions from the online phase that we need to discuss and decide, whether to keep it or whether to change it, to tweak it. So this was a really valuable part of the network verification challenge because we could really discuss with the people who are really interested in these subject areas. And then the last part is to then make those changes, have version 2.0 networks, and putting those back on the website. So those are now on the website with all of those changes, and they're available to download for anyone to use. And for NVC2, we are now in this online phase where people are contributing, and then we're going to have that in-person discussion on a select group of networks that we choose for next year. So just to give a summary of the first NVC, this was a 5-month challenge. There were 50 networks, and people added 885 new evidence, 2,456 votes, 451 new edges. So definitely the most common action was voting. It's the least amount of work. You read the sentence, you review the literature, and then you hit approve or reject. Adding new evidence and adding new edges takes a little bit more time, so there was less activity there. But those are really valuable edges that people added because, there's tons of literature out there and there's something that we're bound to miss. So this is another reason why we're using crowdsourcing to try to fill in the gaps that we might have missed when creating these networks. There were 150 participants, and these participants were from academia, from industry. A lot of students, graduate students and postdocs were interested, people who wanted to get more into network biology, more, people from the lab who wanted to do more computational biology. Those kinds of people participated, and they're from 18 different countries. A lot of people in the US, a lot of people from Europe. Now we're trying to recruit more people from India, China, and other countries that didn't participate last time. And these are the different types of networks that we have divided into those subject areas that I mentioned: cell stress, cell fate, cell proliferation. You see that there is some physiological signaling, different stress networks: ER stress, oxidative stress, xenobiotic metabolism response. There's canonical signaling like mTOR and MAP kinase. And then there's cell-specific signaling like neutrophils, T cells, macrophage. So really there's many different categories that you can choose to look at and verify. And we had certain boundary conditions of the network. So these networks are lung-focused and also COPD, chronic obstructive pulmonary disease focused. So we tried to, only include edges that might be relevant to lung and COPD. Of course, there's always exceptions because, there's always a limitation of literature. So not every single edge in the network is, comes from a lung experiment. You have to make these leaps that, some of the signaling is conserved between different tissues and cell types. And for canonical pathways, for example, we might have included cell lines that weren't lung. And most of it was human, so 74% of the networks were from human literature, but we also included mouse and rat, which BEL has the capability to capture and we have a homologized system that can homologize genes that are homologous across these 3 species. Okay, so this is just an example of what one of the networks looks like on the website, and by clicking on these edges, you could vote on it, you could add new evidence, and you can create new edges. And so now I can just go through, walk through some of the tools in the website and some of the views. So if you would like to participate, you can go to the SBV Improver website and register there. And then there is a different Bionet site, which is bionet.sbvimprover.com, where you can look at these networks and download them if you'd like. This is just a representation of all of 50 networks. So this isn't actually a network to verify, but it just gives a visualization of all the networks and how they might connect to each other. And then you can click on each of these networks to actually delve into the network. So if you click on TH2 signaling, this model opens up, and these are all the nodes and edges that make up the process of TH2 signaling. And there's different search options where you can go to the nodes button here or the edges button and search for specific nodes that you might know to be involved in TH2 signaling. You can check if a certain IL-4 cytokine involved in TH2 signaling, if that's there and what kind of biology is around it. And when you click on one of these nodes, this is IL-4 here, you can see all of the relationships that's connected to IL-4 and see if all of that biology that you know is actually captured in this network. And if it isn't, then you can add your own edge. When you click on an edge, it opens up, first of all, how many evidences are associated with this. So there's 2 evidences listed here. And when you click on the evidence, you see this literature citation that supports this relationship of aldH 3A1 decreasing this chemical. And then you can decide whether to approve or reject it based on reading the literature and based on what you know about TH2 signaling, if this really is relevant for the network. You can give your comments and vote. So here's an example of if you'd like to approve this edge, STAT1 increasing IL-1B, you can give a comment about why you're approving it, or you can decide to reject this edge. Here, there's a comment about transcriptional activity of a factor can directly increase only the transcript of another protein, and protein abundance can be modulated by transcription factor only indirectly. So this is saying that it really shouldn't be a direct edge, it should be indirect, which is just decreases. And then you can also add evidence to an existing edge. So if you read a paper where STAT1 increases IL-1B in another context, then you can add that literature reference here using this form. Same thing with adding a new edge. You can add new biology by clicking on this new edge button. And from the literature, gather that statement, convert it to BEL. Blink BLNK increases the kinase activity of BTK. So this sentence says that, but in order to make that into a usable, computable statement, you would convert that to BEL and put in all the supporting literature in the context. And this is really the curation of what we've been doing for the past 10 years of creating our knowledge base. And there's also a way to automatically generate a BEL statement. So Brett mentioned before this converting text to BEL, and this is a tool that was developed by the Fraunhofer Institute in Germany. So they're one of our collaborators that uses BEL in their research. And so you can simply paste in text from the literature, and then it will send you an email of all of the BEL nodes and the BEL relationships set into text. It's a beta version, so I would strongly recommend to look that over and make sure that it got the relationship right, because some of you may know, text mining is still being worked on, and there's a lot of language issues where it's not exactly apparent to an automatic program what the relationship is. So keep in mind that this is a beta version. So here's an example where you can paste this text in, and then you get your converted BEL nodes and edges in an email. And then just to see, the recent activity, this is a good page to go to, the community page, and you can see what people have been curating and which networks are of interest to them. So here, this is the angiogenesis network, the ECM degradation network. So different people are adding, a day ago this was added or this was voted on, and then you can, from that page, vote depending on how valid you think that is. And then there's this leaderboard. So this is a reputation-based system where you get points for different actions, and then you're ranked according to your activity. And so this is where the invitation to the meeting is the incentive for the top 10 or 20 users. So there's different point systems. The more work you put into it, then the more points you get. Voting has the least number of points, and adding new edges have the most number of points. So this is how we're trying to incentivize the community to contribute. Okay, so just an overview of what we've been doing with. We've been giving talks at different conferences to try to promote this because, of course, we need a crowd in order to do this, and we need people to be interested. And so I just gave a talk. Brett and I were over at ECCB, the European Computational Conference, in Strasbourg last week. And so there were some interested people who asked a lot of questions about this and who are hopefully going to participate. We're giving this meetup. We're also giving a webinar in a couple weeks, and that'll talk more about the applications of these networks. If you're interested in how do we use these networks, and Brett will give an overview of that. And April 2015 is the key date where the open phase will end. So if you want to participate, if you want to move up in the rankings, then it's good to start now, so you can have plenty of time by April. So just to go over the advantages of participating, it's engaging with researchers from around the world. It's using BEL in your research and collaborating with other scientists. You can learn new biology. You can download these networks and contribute your own knowledge to these networks. And so, if you are a biologist, or if you know any biologists that would be interested, then send them to the website. Okay, and that's all we have today. So if you have any other questions, and feel free to ask. Yep.
So any success stories yet? Have you guys improved something to the point where it was worthy of a publication or a new discovery of some sort? Somebody discovered something they wouldn't have with the crowdsourced network?
Yeah, that's a great question. So we have put together some publications in terms of the outcomes, and we actually just last week we got the reviewer feedback, which was very positive. We can't really say if there was some groundbreaking achievement, because all of this has already been found in the literature. So it's not that we're finding new biology, but we're putting it together in a way that people can then use this for their own research. So it's yet to be seen if this is going to be, what kind of groundbreaking breakthroughs there are. But one of the outcomes from the NVC, the first NVC, was that we were able to add more biology. So we gave some examples in the papers that we put together. One of them was, the immune cell networks were very popular among the participants, especially the neutrophil signaling network. The most activity was done in that network. And there was a lot of biology added that had been missing. There was a lot of interactions with other cell types that we hadn't originally put in. So I think it just became a more comprehensive network to capture more biology. Same with the T-cell networks. We decided to merge Th1 and Th2 cell signaling since there's so much interaction between those two cell types. And so, it contributed to a more, hopefully holistic view of what happens in COPD biology. Any other questions? Great, thank you so much for your attention, and feel free to sign up if you're interested.